With the emergence and rapid spread of new coronavirus variants, the urgent need to better detection and tracking of these variants has been recognized and prioritized. And of course, genomic sequencing is the core method for this and for establishing a robust COVID surveillance network. We have been privileged to work with the researchers at LabCorp starting in April of last year to develop a high throughput workflow for complete SARS-CoV-2 viral genome sequencing. LabCorp then received a contract from the CDC to apply this pipeline for a large-scale longitudinal genomic survey that aims to improve understanding of these mutations, how they are transmitted, and how the public health response to these mutations can be improved. Through this program, the CDC aims to more than double the rate at which it conducts genomic sequencing of the COVID-19 virus. Details about the sequencing pipeline are shown here, using 1.2 kilobase amplicons that span the entire viral genome. Pools of up to 900 samples per SMRT Cell are sequenced on the Sequel II or Sequel IIe systems. This effort has already delivered impactful results. For example, PacBio sequencing was used to identify the first instances of the more problematic variant from South Africa, first in South Carolina in late January, and subsequently a few days later in Maryland, Virginia, and only a few days ago relative to the time of this recording in the state of North Carolina. This highlights the power of partnering complete, high-quality viral genome sequencing with LabCorp's national network of sampling, allowing the detection and the evolution of new viral strains in time and space in the US. The pie chart shows the volume of data uploaded to the GISAID database during the week of this recording, with PacBio entries accounting for about half of all the entries. A detailed end-to-end protocol describing the PacBio Hifi viral for SARS-CoV-2 workflow is now available to allow anyone interested in implementing this real-time surveillance workflow to do so. We believe that highly accurate long reads offer critical advantages for surveillance, and I've listed 5 aspects here which I would like to comment on in a little bit more detail. First, through the use of longer 1.2 kb amplicons, the PCR reactions are simplified while also providing more even genome coverage and striking a good balance relative to the integrity of the viral nucleic acids in the samples. Second, with fewer amplicons, primers anneal to less of the viral genome, lowering the chance that new viral mutations will cause dropouts due to primer mismatches. This is shown here in more detail. The PacBio workflow only uses 29 amplicons, corresponding to only 4.5% of the viral genome covered by primer sequences. This compares favorably to the Arctic primer set, for which almost 1/5 of the genome is covered by primer sequences, or the Ion Torrent workflow, which has priming to about 40% of the genome. So with that workflow, there's almost a 50-50 chance for a new mutation to fall into a primer region. And third, 99.9+% Hifi reads from longer amplicons allow confident detection of all variant types, from SNPs to up to 300 base pair indels, which have been seen. And they allow for phasing of variants in samples that contain multiple subtypes. This is shown by example in the next 2 slides. The first shows 2 examples of 2 different deletions of approximately 300 base pairs in length in both cases. In both of these cases, these are completely contained within the 1.2 kb amplicon. In contrast, they would interfere with the PCR of amplicons from the Arctic set. The second example shows the clear presence of 2 distinct viral subtypes, with several variants present in this approximately 2 kilobase region. Through full-length Hifi sequencing of the longer amplicons, these variants are automatically phased over long distances, thereby allowing the confident identification of 2 distinct subtypes. This is something that would not be possible here with the shorter Arctic amplicons. In addition, because each Hifi read is extremely accurate, coverage of only a few reads is sufficient to very confidently obtain a high-quality consensus and make these variant calls, and this also helps with pooling and amplicon balancing. From a workflow perspective, the protocol is built to allow for flexible batch sizes and can be scaled to sequence from as low as 48 samples up to 900 samples per SMRT Cell. The high level of multiplexing makes this very cost-effective, so per sample costs are very comfortably within the reimbursement rates for complete viral genome sequences. To summarize, we believe PacBio Hifi sequencing is a very powerful technology for enabling robust pathogen surveillance. And while today's efforts and focus are on detecting and tracking COVID-19 in our communities, the infrastructure that is being developed will bode well for our preparedness and sophistication of fighting COVID-19 and other pathogens in the future. So getting smarter on understanding the impact of COVID variants on vaccine responses, patient prognoses, and then longer term to quickly identify what pathogen is responsible for respiratory infection and what strain it is, whether it is bacterial, viral, something known, or something new, will be possible. We are looking forward to working with you on implementing these solutions, so please reach out to us if you're interested, either at our COVID landing page shown here, or you can also use this QR code to get in touch with us.